Thank you, Chair, and thank you, Hans-Carl, for the introduction and the presentation. I will continue with the clinical issues concerning this non-clinical clinical guideline. The concept paper promises that we will address these topics, and I believe that you will find some refinement or a new text on these topics in the draft guideline. But in the interest of time, I will not go through all of them. I just pick up a few issues related to clinical trials. The first thing is the selection or choice of efficacy endpoint for the efficacy and safety trials. And maybe needless to say to this audience that the aim is really to detect differences, not to demonstrate efficacy and safety as such again. And that changes the criteria for selection of the endpoints, which may in certain cases may be different from those endpoints that we use for new active substances. And the main criteria for selection of the primary endpoint for these studies is sensitivity to detect relevant changes. But of course we expect that there will be also other endpoints that are in common with those endpoints that were used for the innovators' pivotal studies at the time of the licensing. This would ensure the assay sensitivity. Comparability margins are difficult to describe. We make reference to the relevant guidelines, ICH guideline and CHMP guidelines. So basically these margins should be defined by statistical and clinical criteria. The problem is that we don't often have very much data from the originator that could be used for this purpose. But this is the principle anyway. Then, as you may have noticed, the current guideline, the old one, doesn't speak about equivalence or non-inferiority at all. But very soon after the publication of this old guideline and by the entry of the biosimilars and increasing amount of scientific advices, the CHMP and the Scientific Advice Working Party very soon took the position that the design should be equivalence design. And this is reflected in our current guidelines other than the main non-clinical and clinical guideline. Now we say that the primary expectation is that the design is equivalence, but there might be exceptions. We know that in certain cases the equivalence design would lead to unreasonable large studies, so it's not practical. And the way to propose non-inferiority design is to give us justifications. And you may wonder why we always need justifications. Don't we know by ourselves what we need? But the science is evolving and we keep getting new information, and therefore we always ask companies to come up with justifications. Basically, what we wish to know: are there such characteristics of the reference product that might allow or prevent the use of non-inferiority design. These may be related to the safety profile, tolerability, the dose range, especially the dose response within this range. And on the basis of these criteria, the companies should be able to convince us that increased efficacy would not be expected. And when we talk about increased efficacy, it's not the efficacy we are so much worried about, it's the pharmacological action that may lead to some adverse events. So this is an opening which is in line with some other guidelines in this field. Nevertheless, it is advised that the companies will consult the regulatory authorities before embarking with the studies. And then finally we have this difficult issue of extrapolation of efficacy and safety from one indication to another. And we try to a little bit open our thinking in this issue, and we explain that we again ask for justifications for the same reason as I mentioned, based on clinical experience derived from the literature. We are interested to see the mechanism of action of the active substance. We of course appreciate data on the pathogenetic mechanisms involved in particular diseases, but crucial is the mechanism of action of the active substance. These two issues are often mixed. It is extremely important to know whether we have the same receptors involved in all therapeutic indications, because sometimes we might have several related receptors cross-reacting with the therapeutic agent. And it may be that these justifications are not sufficient, and we will need additional data. This would be the case when we have different active sites in the active substance, especially monoclonal antibodies, where we have difficulties in defining what is the relative importance of the different sites in different parts of the molecule. Then we would have to get some additional data. We need to know about the receptors, as I mentioned, whether they are the same in all indications, whether there are receptors that have a limited distribution, different affinity, and so on. And then we need to consider the safety profile of the active substance or the product in various indications. There might be differences in safety or efficacy. Now, the additional data certainly doesn't mean new randomized clinical trials. That might be the ultimate case, but usually I think we can get some assurance from the non-clinical in vitro functional testing. The tests can be improved and modified to give some more information. And of course, we can also look at the clinical pharmacodynamic endpoints. So I think I leave you at this point and save time for the discussion. Thank you very much.